one. We are now live.
Great. Thank you, Sabrina. Really appreciate this. And thanks everyone for joining us today on a call we're having, an interesting call, one of the more interesting ones that we've hosted in a long time, computational biology and drug discovery. We have with us the GAN Therapeutics CTO. GAN is a company that we cover. Javi's with us. We have two experts who actually have a lot of expertise in this area. We spent a lot of time gathering up these three folks and getting them one call together. Brandon Allgood is PhD. He's got a degree in computational physics, right, Brandon, if I'm getting that correctly, and has been involved in starting many companies in this area, and that's just putting it very mildly. Jason Paragas, Jason, I hope I said your last name correctly, is also a PhD and was with Lawrence Livermore from 2012 to 2019. I don't know why he chose to leave the land of the gods for us lesser folks, but here he is. Thank you all three for being with us today.
Thank you for having us.
Great.
Our pleasure.
Thank you, everyone. So we'll keep it, my goal will be talk as little as possible. Just a couple of housekeeping matters. Anybody has any questions, put it in the chat box, or you can send me an email at hartaj.singh@opco.com. Again, that's hartaj.singh@opco.com. And then we'll leave some Q&A towards the end. We'll probably spend a little bit more time with the KOLs in the beginning for the first 10, 20 minutes, and then bring Javi in to give us GAN's perspective. But maybe, Brandon, I'll start off with you. At a very, very high level, computational methodologies in sort of drug discovery and drug design, those are almost two different things. But in drug discovery, let's talk about that. How do you sort of define that at a very high level? And Jason, I'll ask you the same question after that, and then we can kind of start drilling in.
Yeah, so I think you start with the term that you use to describe the field, drug discovery versus drug engineering. So I would say that the definition behind computational drug engineering is what I like to say. That's how we just separate from what I would say is a more traditional approach. If you look at the last 70 plus years and the ways in which the vast majority of drugs that are on the market today were discovered, it was through massive efforts, largely based on serendipity, or larger and larger, what I like to call larger and larger shotguns. Right. We built an industrialized process that went after, not to diminish the things that we've achieved, but go after, I would say, fairly simple disease, inhibit an enzyme, lower a biomarker, make billions of dollars. Right. I think what we've done with this large industrialized machine is really kind of pick all the low-hanging fruit. And what we now have in front of us are more complex diseases that are not ones that are singularly focused, but that are broader and often in many cases syndromes. And in these cases, developing artificial in vitro assays becomes very limiting in our ability to approach these diseases, which means we need to go to more complex systems, which means with those more complex systems, we need tools that can understand and analyze those complex systems. And that's generally where those computational methods come in, both in terms of machine learning as well as simulation. So I would say my definition is really being able to bring data in and make data-driven decisions through computation.
Yeah, no, that's fantastic, Brandon. And Jason, just kind of going over to you. I mean, you both have seen the progression of this. I mean, I was in the 90s doing some work in neurobiology, and there was, even at that time, like a dream, of doing drug discovery and using computers to help us. Now it's almost 30 years later, and there's been progress, but I call it a little bit patchy maybe from my perspective. But your thoughts on that? How do you define it, and what do you think of the progress over the last 10, 20 years?
Well, I'm very excited about the progress we've made. If you really think about what biology is, it's really the most complex system. We're trying to go from analog to digital. We're talking about, orders of magnitude more complex problem than we've ever had to calculate before. If you think about, cars and safety, those crash test models, they've been around for a couple decades, and we're now with cars going to a concept of zero fatalities, and that's really enabled because of computational simulations, right? But again, that's a physics-based approach. We know all the variables, we know all these sorts of stories, right? It's orders of magnitude less complex than biology. Now in biology, finally, there's a kind of, we're at an amazing convergence point. One is we're measuring biology at scales that we've never measured biology. So if you did a clinical trial today and said, we need to do a whole genome sequence on all the participants, no one would laugh at you. They would say that's totally reasonable. So we can measure the scale of biology up and down, left and right in an amazing way. The second thing is the tools. This is what we're talking about with AI, whether they're simulations that GAIN is doing, generative learning, graph, quantum, all these different tools, all these different data engineering tools that Brandon was talking about. They're emerging and they're really important. Not one tool solves all problems, but certainly the platformer tools or the portfolio tools is amazing as we put them together. And the third piece is the technology or the hardware. If you think about today's frontier computers, what we call the exascale machines, they are finally getting to the point where they can really start to calculate biological complexity. So we did a calculation a few years ago when I was at Livermore, and we said an exascale machine, which is the current fastest machines, and just to give you kind of what this means, an exascale machine is about 1 billion iPhone 14s, roughly, running in parallel, somewhere around there. But an exascale machine now probably has the complexity, has the power to calculate 5 years of heartbeats in 5 minutes. And that's really, that's starting to say you can do something impactful with medicine. So we're really just finally hitting that point where the tools, the data, and the hardware are converging, where we can really make this meaningful thing against the most complex problem. It's harder than astronomy. It's harder than physics. It's a hard problem, and we're finally there because the pieces are finally starting to emerge.
Yeah. And Jason, just to follow up on that, before I just
Sure.
want to go to Javi, because Javi is a professor at the University of Barcelona, so I'll bring him here real quick too. But Jason, you mentioned tools, data, and hardware. Where does software stand in all of this? I guess it's the analog of the wet part of computing, right? So I know we're going to get into that more, but where do you put software as within the tools category, or or connected to hardware?
It's both. It's both. It's the tools, it's how we measure biology, whether it's a human genome, a sequencer, but it's also whether it's a generative learning or a simulation. It's a software tool that will give us the answer. So it's really both.
Yep, got it. And then Javi, maybe just if we could get your opinion here. I mean, you're a professor. Aside from GAIN, what is it that when you look at computational methods, in drug discovery, how have you been sort of defining it? As you're out there teaching your students and doing research, what are you seeing sort of more from the academic side than really from a company side?
I mean, what you see is some methods that actually were developed some time ago now become not really useful because of all these advances in computation that you see in the software and in the hardware. And it was before, doing my PhD, to study one molecule, one particular system, you would have to work a very long time just setting it up and then waiting, weeks or months to get the calculation done. Now you can do that, preparation in almost one click, all the results overnight. And that means that you then kind of scale it to, a much larger set of molecules. And you have to consider, many molecules. So that's what we do. And we can do it at scale now. And on top of that, obviously, all the methods of the theory has also improved. So we get results, orders of magnitude faster and orders of magnitude better than we used to. But it's a phenomenal technological and scientific development that we have seen in the last 20 years.
No, that's and that's actually, you know, we're kind of seeing that here on, you know, as analysts with companies kind of coming through and utilizing these methodologies more and more, and actually getting credited for it, you know, in the public markets. I mean, we're not private, you know, company analysts. I mean, I'm a public company. You know, it's a different kind of way of looking at it. And then maybe Brandon, just kind of pivoting back to you, you know, just still talking maybe on a higher level. You know, if you can just kind of highlight, Brandon, what do you think are the most recent, the recent innovations in sort of drug, you know, discovery that really stand out to you over the last 5 to 10 years, whether they're company specific or research specific? What are the one or two that you think have really sort of opened your eyes from a drug discovery perspective?
Yeah, I would say over the last 5 to 10 years. Now we're going a little bit far back, but certainly, I mean, the one that I would say is the top of the list would be the generative work around AlphaFold. So obviously around protein structure prediction, that's been a grand challenge for many years, and for, you know, Google Brain and the others to achieve that is a real, you know, I think a sign that, you know, we're starting to really move the needle in terms of generative AI. And again, I think both to what Jason and Javi were saying, you know, it took hardware, it took software, and it took data in order to be able, you know, it took all three of those advances in all three of those to get there. I would say, you know, beyond that, we've got, you know, again, if we're going to go back 5 to 10 years, obviously deep learning. So, and especially in small molecule design, you have graph-based machine learning technologies. So this has made a huge leap in our ability to not just predict activity, but predict other properties. So again, in my mind, especially small molecule drug discovery, it is a high dimensional optimization problem within a discrete space, right? So it's not just finding compounds that are active, but compounds that are active that can get into the body, that are not metabolized too quickly, not metabolized too slowly, have the right properties, right? And so there are a lot of properties where I think you know, beyond activity, where a lot of these graph-based methods on the molecules themselves, so message passing graph-based methods that have come out of places like MIT and Caltech, have really been, I think, advancing our field and our ability to predict. And I would say, you know, I would say the scale of the cloud and the scale of just computing on its own is enabling, as we both heard from Javi and Jason, just being able to reach that scale in terms of, you know, being able to light up 100,000 GPUs or CPUs, run these very large simulations, be able to collect that data and sort through it. That's been hugely enabling on top of everything else. And then data. You know, one of the things that you know, I think we often talk about is, you know, how is this field different from other fields? And I would say the biggest difference is data, and our accessibility of data, amount of data, cleanliness of data. All three of those things have vastly improved over the last 5 to 10 years and have been hugely enabling. We're not done yet, don't get me wrong. I mean, most data sets are still pretty bad. They always say that data is the new oil. It's dirty, it's messy, and it's not really well contained. So I would say those, if I reflect back, there's a few achievements within the machine learning community, but it's really also kind of these other pieces that need to come along and have come along to really push us forward.
Yeah, no, absolutely. I mean, I can't give you any specific examples, but as an analyst, you know, when we vet companies, look at public companies, you know, cover a company like GAN, we're seeing the pace of computational methods pick up. And sometimes we don't see it directly. We see it in an indirect manner by just companies producing more, you know, very good, you know, especially small molecules, you know, at scale, getting them out more and more into the clinic, you know, from discovery, from research into preclinical and into the clinic. Jason, you know, I guess I'll ask you the same question with a twist. You know, you've been,
Sure.
You know, also been a venture capitalist, you know, for the last 2-3 years. You see things on that private company side. You're looking at it from the point of view of getting, you know, an ROI on very, very, you know, more risky kind of endeavors, right? Earlier stage. What have you seen from a computational perspective that really excites you, with that business angle, over the last few years, I would say since, over the last 5 years, essentially?
Well, I think, the big concept that I'm always looking for is closed loop. Can you connect the laboratory to the computer, really, or the patient in a closed loop sort of thing? I think that is, there's a few companies out there that are doing that, especially on the private side, and it could be very disruptive. The second place, we've been looking at generative for a long time, has really been in looking at these hybrid-like approaches. So, you may look at some applications in quantum. You may look at some applications in graph. You may be looking at some applications in simulation, and you put them together to learn a model that's outside of the data set you started with. And so I'm really excited about what's going on around generative and these different approaches being weaved together to kind of give you an answer that can exist outside of what we've measured today, and then obviously you have to test it. I think the third space that I'm really excited about, and we've done some work even when I was at Livermore, is these language models. So we've been awestruck by ChatGPT, but the reality is these language models have been around for quite a while, and they've been applied to biology for quite a while. And in fact, some work at Livermore has designed some antibodies using these language models already, and it works pretty well. So I'm really excited about what's going on with the language models, because it's, again, it really is accelerating the pace, really taking advantage of scale, and starting to really, if you the complexity of biology, you're going to need more than one approach to get to really change the game for how we develop drugs.
Right. No, absolutely, Jason. And then, Jason, just to follow up on that, I guess, from the point of view of, as an analyst of public market companies, and thinking about, a lot, our clients who are on that end, when we look at companies on the private side, let's say you've looked at them, you funded them, what are important, questions we should be asking? Is it really just all about scale? Are you interested in, a problem being solved and then how you scale it to get to public markets, or how to think about that?
That's a great question. It's a combination of things. It's really, what are you designing the code to do, right? We can say, oh, we're going to use machine learning. That's great. What problem are you solving? Is it solvable, right? Do you have the data set to solve it? And then how fast can you really validate these predictions that the machine will make, or these forecasts the machine will make? One of the things I've seen again and again and again is that the computational team will put out 10 hypotheses, right? And 9 of them will be wrong. And everyone will jump up and down and say, I told you it didn't work. Right. But the reality was, one, you're only dealing with 10 hypotheses, right? Or 10 molecules, right? Instead of, 10 million. And the other thing is that one, you probably would have never found anyway. So it's that team. Can the team do it? Does the team have a plan? And is the team going to get you to that vision, the problem that you're going to solve, not the tool that you're going to use, but the problem that you're going to solve in biology? And that's really what I would look at.
Yep. And Jason, I'll just ask you a follow-up question here before going back to Brandon, which is that, you look at DeepMind, right? And you look at all of the protein models, and I guess there's another company, I'm forgetting the name, Rosa, Mind or something. There are a couple of companies doing this, but DeepMind is most well known, Google. And it's interesting to me because that seems to have real business implications and probably could be a, cash generating business for Google, sooner rather than later, right? But the world is fascinated with ChatGPT. I was blown away by DeepMind, these protein, 3-dimensional protein. I mean, is it just, I mean, what are your thoughts there? Why is the world not more excited about that? Why are investors not more excited about that? Is it just because Google is providing it for free? Like, what exactly is going on there? Because I think it's fantastic.
I agree with you. I think it's absolutely fantastic. It's a technical milestone. And they did the right thing by really making it accessible. But if you think about what we're really trying to do in health, what Google did was put one brick in the foundation of computational drug design. And so protein structure, absolutely an amazing achievement, but it's only one brick. The second brick is we need the gene regulatory pathway. So that's how these proteins line up in kind of a telephone telegraph and they transmit a signal. Can we do that in the computer? Almost, maybe, not yet, but we need to be able to do that. Then can you put it inside of a cell? And can you put that cell inside of a tissue? And then can you put that tissue inside of an organ? And then can you put that organ inside of a person? Okay, so what you see is that it's an amazing technical achievement, but it was only one brick in the foundation of where we need to get to in the next 5 to 10 years.
Yeah. No, that's a really good insight. I mean, that's the foundation of many different private companies, right? That hopefully as public investors will be helping either take to the markets or covering in the markets.
Oh, yeah.
Brandon, I guess with ChatGPT, and I have a personal connection there with a friend that works at OpenAI, and I respect him tremendously. And by definition, I have respect for that company because I think he's one of the smartest people I've ever met in my life, and he enjoys working there. What are your thoughts, not on ChatGPT, but has it actually raised the level of awareness, more chatter around, drug discovery, computational methods of drug discovery, or not really? You haven't had that halo effect yet.
I don't know that it's raised the bar in terms of exposure of drug discovery and development. I will tell you that every single CEO I have spoken with in the last 2 months wants to know how GPT-4, ChatGPT will, how will that affect drug discovery? And I don't think there's a CEO on the planet that's not asking their CTO or their engineering team, how can we use this? How is this going to change things, right? And I think it's so in some ways it's definitely bringing bringing up the topic of machine learning and how that can be used in business in general. But I don't know that it necessarily going to raise the level of computational drug design. I think there in kind of smaller circles. Certainly, I think it is showing that generative is becoming a force versus just versus your typical supervised machine learning, or so I think there you are starting to see, at least within biotech, within pharma, you're getting more interest in and more want to see these generative methods. So I think in that case, I think it is definitely kind of raising the level of awareness and capability within at least the generative space.
Yeah. And then Brandon, I'll just ask you one question before going to Javi and talking a little bit about GAIN. But when you look at the private and public companies out there, Brandon, that are kind of in biology, that are using some of these computational tools, without talking about a specific company, because I don't want to put you in a bad spot, but what tools or what areas really excite you when you think about whether it's private or public biotech companies in drug discovery?
So yeah, I think again, to one of the things, I mean, Jason hits on great points, right? So one of the things that I find exciting in what people are doing is this closed loop. One of the things that people forget about, even in our field, is that there's a basic tenet of machine learning, that the training set is pulled from the same, your test set is pulled from the same distribution as your training set. What that means is that the data that goes into building the model is supposed to look very much like the data, like the things you're trying to evaluate it on. Now, the thing in chemistry, again, what we talked about, we're always extrapolating outside of that distribution. We're always trying to go for that, find new chemistries, new modalities. And so by definition, the models are not appropriate right away. And so this is where the things like Jason was talking about, where you need to have that loop, you need to have that connection between the laboratory and the machine learning, because you're always going to be in an active learning loop. And one of the things that I think we'll probably touch on a little bit later, and I believe, GAN's thinking about, is one of the ways in which to speed up that loop is kind of short circuit it with simulation, right? So you can stay in silico a little bit longer and get better extrapolation before you ultimately come out. Because computation, let's be clear, one of the biggest drivers behind machine learning is the fact that CPUs are cheap and storage is cheap, right? That is literally that is the single reason we are where we are today. And so if we can stay in silico for longer and get further along before we go into atoms, because going into atoms takes, synthetic chemistry, all of these other things take so much longer and don't scale like compute does. So I would say that's the thing I'm most excited about, is to see those systems that are connecting the lab, connecting simulation, and connecting machine learning in a really integrated way.
Yeah. That's really good, Brandon, and thank you, to both of you. I mean, for kind of setting the stage for us, and maybe we can just go to Xavi here, who is working with a company, a chief technology officer that's actually working on the front lines here. Xavi, maybe you could give us a quick idea, in about sort of 3 to 5 minutes on what it is that GAIN is doing roughly, but more specifically the computational aspects of what GAIN is doing and what makes it unique.
Yeah, so I really want to thank Jason and Brandon for and I really loved what they say and learned a lot as well. And one of the things that I think is key is that you have to know what problem you're trying to solve. And in our case, what we're trying to do is not pick up these lower hanging fruits, as Brandon was saying, but go for a new class of drugs, which are allosteric drugs that bind to places that nobody know that actually the proteins would like to bind molecules there, and that's very difficult because there's no data. But how you can do that? You can do that with physics-based methods. How can you speed it up? By having these active learning methods, right? That you have the physics, then you feed it back onto machine learning models, so you can do things at a scale better, right? And we've been talking about computation, and it's really amazing and very exciting times. We're having a lot of fun with all these generative models for 3D, generating 3D structures, or the methods that predict things that seem simple, like crossing a membrane, whether a molecule could cross a membrane or not, is not so trivial, and it's really important for drug discovery. But then, of course, there has also been advances in other fields of computation, like the physics-based methods, and also in biology. Biology is incredible, like all the high throughput methods, like all the omics that we know, the proteomics, the genomics, RNA-seq, are really so commonplace nowadays. But also high content analysis, the fact that you can generate really much better models, right, with iPS cells, right? So these pluripotent cells, and then try to what is fun and also difficult is try to bring all the tools to the problem. And our problem is, can we identify molecules that bind to these allosteric binding sites, make the proteins function differently, rather than just break them down, which are other drugs do. You want to modulate it, sometimes gain a function. That's the name of the company where it comes from, and then actually see that it really works, right? And you want to make use all of the computation. This really the interface between the chemistry and biology, but then finally show that it works on relevant biological systems, and yeah, that's what we have been busy doing in the last few years.
Xavi, and as Brandon was saying, maybe we need to host one of these in person in Barcelona, because I imagine it's a lot of fun doing it in Barcelona. We're in New York City. I'm in New York City. New York City is a lot of fun too, but right now a little too cold still. I imagine Barcelona is better. Javi, one thing I just want to be clear, before we dive into this a little bit deeper is essentially GANX methodology is to use these three-dimensional like models, right, of these proteins, kind of have them interact with solvents or water in silico, right? So you model that, you find the sites that are active, these so-called allosteric sites, away from the binding site, right, the catalytic site, and then you take it into a petri dish. Is that a simplistic way of kind of explaining what GANX is trying to do?
No, that's what we take the three-dimensional structure of the protein. We use molecular simulations that identify where the protein would like to interact with organic molecules. So we identify these allosteric binding sites that are what we call it a druggable, or some people call it ligandable, that really want to bind small molecules, and then we identify molecules, and here is where we really try to make this discovery problem into an engineering problem, right? As Brandon was saying at the beginning, and we have been very successful there, right? That not I wouldn't say we're there yet, right? But in terms of finding molecules that really match, complement the protein, and then give you a signal in the primary assay, we are really satisfied with that. It's just a very high success rate for a very difficult problem. And we don't have to do thousands of compounds to actually find molecules that are active. Just a few hundred molecules, and we find molecules that are active. But that's just the start of the process, right? So we start something that nobody else we think a few other people can actually start the project so efficiently. Now everything else has to come together, right? Finding the right molecule, optimizing it, make sure they have all the properties that have been mentioned before, and then actually check that you have the right biological response, because biology is, that cannot be understated. The complexity, as was said before as well, the complexity of biological systems is just, is something that is mind-blowing. And we try to predict, and we try to suggest how biology is going to react. But in the end, you need the right biological system on which to test the molecules, get your readout, and then make something that is you can extrapolate to what will happen in a patient. And that's definitely a frontier still.
Yeah, no, absolutely, Javi. And that's one of the things that excites us about GAIN. But, Jason, maybe just pivoting to you talked about putting a brick in sort of, I'm forgetting the Pink Floyd song. I should not be aging myself.
The wall.
In the wall. No, I knew it was in the wall, but I didn't know whether it was specific wall or not, or whose wall it was. But, you talked about a brick, and it seems like Javi and GAIN, are tackling one or two bricks, in that wall, a very important couple of bricks, right, to kind of speed up and scale up drug discovery, and hopefully have higher, greater precision. But what would you think, Jason, would be a next logical step, the next brick and the brick after that? Would it be prior to the process that GAIN is doing? Would it be downstream? How to think about that, using computational methods?
Well, I think, GAIN has established kind of one of the cornerstones of the way you go. So, obviously they're looking at a protein with an allosteric inhibitor through simulation. So as we computational approaches and hardware advance, right, do you start looking at protein complexes, right? Do you start looking at signaling, right? Take a classic like RAS, right? KRAS. Right. And you say, well, look, does this influence signaling or function, right? Right now we would have to go to the lab and say, okay, does this allosteric inhibitor work in the lab? So, but eventually, kind of to Brand's point, can we hold in silico longer function rather than just the binding, the interaction, maybe looking at it at a single protein level? Can we start getting at more complex biological systems?
Yep. No, that actually helps a lot. And then Brandon, just before going to, I just got a question, Javi, on from one of our listeners is, And the specific question is, I'll read it out. Can you use AI-generated protein structures like those from AlphaFold in GANX drug discovery platform?
Yeah, absolutely. So wherever the structures come from, they all come with some limitations. They can be experimental structures, like in X-ray crystallography, or they can come from cryo-EM, or they may come from homology modeling, or they may come from AlphaFold. And what you have to do is to understand the methods that were used to generate those 3D structures, and then analyze those structures critically, wherever they're coming from. And then, so that's what we do. And then when we check that actually the quality of the structure is good, then we can work with that structure, regardless of where it's coming from. But there's a sanity check that you have to do in the beginning, and you have to do it, I mean, for any method, right? You know, experimentals, when I was going to say kid, but with the first time I actually look at the 3D structure, it was, I thought that was, crystallographers were like the god coming with a 3D model of a protein, and that was not right. There's actually the wrong things in any 3D structure, so you have to be very careful. But
Yep, no, Javi, that makes a lot of sense. Whenever I look at an X-ray crystallography structure, it's kind of like looking at the stars. I'm like, oh, look, somebody saw a serpent up there. I'm like, what were they smoking? Because I don't see a serpent at all. But apparently, if you connect 4 stars, it becomes a serpent. Same thing with crystal X-ray, methodologies. But Brandon, I guess I want to ask you a question here. Is that you're sort of working, and sort of the head. You're founder and board member of the Alliance for Artificial Intelligence in Healthcare, right? And you interact with a lot of folks, right? You're interfacing, I imagine, with a lot of different folks, including us on this call. And it seems like small molecules, it's on the drug discovery side, have been really a greater area, right, for computer-based methodologies. Just why is that? When you see from your broad perspective, why is that small molecules, and especially this age-old, using chemistry, right? What the chemists were doing, 50, 100 years ago. Why have computational methodologies been more useful here?
2 reasons that are probably a little mundane. Number one, molecules are small, so they're easier to given computers and the advances of computers in the 90s and the 80s, you could handle chemical structures in terms of encoding them. And so I would say they're smaller. Number 2, we as an industry have been producing small molecule drugs for so much longer, which means that there's more data. And we've developed more methods for generating that data and generating that data at scale. I would say I don't think it doesn't have anything to do with algorithms themselves, and more just around computational tractability and data.
Yep.
If I may add, actually, the other reason, in my view, is that particularly for designing antibodies, you have they have engineered methods that it is like evolution, right? There's just follow, things mutate and then just select the molecules that are better, the antibodies that bind more tightly. So, and there are a variety of methods to do that without having to worry about what's happening. I mean, just a process. And I think what we want to do with this learning method, right? The active learning methods in the computer is try to do the same thing also with the small molecules. That we start with a molecule that's perhaps not the best one, but then do iterations as they do when they mature an antibody, and actually come up with something like that. It has many right properties, and then do the testing as was said before, right? Try to do this process in the computer. So there's more scope for small molecules to do that. But I think
no,
yeah.
John. Sorry,
I would say over time, what you're going to find is again, the small molecule design what has led into this field, but large molecules, RNA, cell-based therapies, all of these CRISPR therapies, they're all going to include machine learning and explode. So again, I think the nice thing about, and again, this is where that kind of hardware, software, data kind of they all reinforce each other over time. These are new methods for generating more data, which then enables better algorithms, which then pushes hardware, and kind of, so you're going to see a lot of these other therapeutic areas catch up in terms of the amount of therapies we have and amount of therapies going into clinical trials, largely because of the ability to use machine learning to generate this more complex data and be able to run it on this larger hardware. I mean, I think Jason laid it out very well in the very beginning.
Yep. And I was actually going to go to Jason, and just ask Jason, in terms of like, it seems like small molecules, where folks are focusing. I mean, folks like myself, right? I mean, it's easier for me to think about GAIN and what they're trying to do. It's within the context of how I envision preclinical and clinical drug development. So they're speaking the same language to me on a certain level. But Jason, what other areas aside from small molecule, what's the next you would imagine? Is it the protein-protein interaction? What other areas do you see as amenable to this computational drug discovery approach?
Well, I think we're going to see kind of an explosion in antibodies and peptides. After kind of in the spirit of supercomputing, Livermore actually designed an antibody about a day and a half after the Chinese published the SARS sequence, and they used computational methods. They had access to a big supercomputer that most people didn't have access to, and were able to design a molecule in the span of hours. Right? We're seeing the same thing around peptides too. The ability to design, and peptides sit somewhere between kind of a large molecule and a small molecule. And so what you're going to start to see is computation following kind of where the frontier of therapeutics are. And probably better than everyone, the generation 2 and 3 of IO therapy. We need it now, and we got to keep running this fabulous advance in IO. And I think we're moving into a more complex environment, and that's just going to call out for us to have these designs of antibodies that induce particular functions, peptides that induce or inhibit particular functions. So really exciting time coming up in that space.
Yep. And Jason, I guess one of the things we struggle with always when we're vetting companies, that are private or smaller, is the difference between, what is a tool, right? What company is developing a tool versus what company is actually developing a therapeutic, right? And there are two very different problems you're trying to solve there. How to think about that from a computational perspective? Like, what would be the advice you'd give us when we approach, issues like that?
You hit onto the hardest problem in the space right now. We can develop platform companies, technology platform companies, like we do with like Salesforce or whatever. We develop a computational platform that can do something. But and they can produce a lot of beautiful drugs, and there's no question about that. But at the end of the day, it's still going to cost hundreds of millions of dollars to get that thing across the finish line. And that's really it's almost a completely different business. And, there is definitely going to be a time when a pharmaceutical company that's been from the ground up designed as a computational company becomes the next Merck, Pfizer, AbbVie, you name it, right? And that day is very close. Who it is, I don't know, but it's a very, very interesting problem. And I think that our traditional view on what is a pharmaceutical company, what is a biotech, what is an AI drug discovery company, I think we're going to come out of this in the next 5 years with something completely different that may look closer to Microsoft, Google, more of these ecosystem companies, and it may be very different than what we know of today.
Yeah, no, Jason, absolutely. And actually, one of the reasons we cover GAIN, and we're having this call, is for self-preservation also. I'd like not to be a dinosaur in 5 years.
It's a real problem.
Like drug discovery in a very biological sense, right? But Jason, before I go to Javi, and I want to ask you and Brandon this in sequence before going to Javi and asking him specifically about how GAIN, one of our theses on GAIN is it's not just that, they're actually, bringing more small molecules into a petri dish, at scale, cycling them through them more and doing the biological experiments, right? By doing these computational methods first for these allosteric sites. But the theory is that, biotech success rates or the opposite failure rates in phase 1, phase 2, phase 3, and actually have stayed pretty consistent in the last 50 years. It really hasn't changed. Studies that were done in the 70s and the 90s, more recently by MIT and stuff, have basically shown the same: 10, 15% phase 1, 25% in phase 2, 50% in phase 3. That stayed about the same. And the hope is with computational methods, you can actually increase that probability of success, right? You're getting that precision up further, which I believe is of value. That will generate an NPV, right? What are your thoughts there? Could that be done, if we're using computational methods as one side effect in the near term to help, grow this nascent field?
Absolutely. I think we've only scratched the surface about the possibility. If we start thinking about it in terms of a pipeline,
right?
I'm sure you guys have somewhere in the back of an office, you've mapped all the computational companies against the pipeline.
Yeah.
And what you'd see is a very skewed distribution toward discovery, right? How many companies predict CMC? Not many. How many, and how many predict patient, patient trial outcomes? Not many. But, we probably have 250 AI discovery companies, right? So I think it's, there's a lot of opportunity in these other chevrons to develop predictions, to develop forecasting, to do those closed loop experiments. So, that when you say, we should all get depressed when we hear 25% success rates, right? But if you're an investor, you can go from 25 to 90. And I know that's a silly number, right? But if you think about Intel, they computationally design their chips, right? And they're usually off by 0.1 of design spec, 0.2, tiny amounts, right? And I think that's the promise of computational drug discovery is at day 0, when we do our design, can we be within 0.1 of spec, not 5% success rates or 10% success rates, right? That is an enormous opportunity to grow the field, make up this difference, and really build the next Merck, Pfizer, GSK.
Or GAIN Therapeutics based in Barcelona.
Or GAIN Therapeutics, that could be it, right?
Then you'll be invited for sure.
Yes. Brandon, if you just want to give your quick thoughts on it before I want to pivot to Javi and just talk a little bit about, what it is specifically they think they do better than other companies out there.
Right. So I love the statistic you pointed out, but if you also then skew it to, how successful are we at producing molecules that perform like we want them to perform in vitro, we're getting better and better and better. The problem is the biology risk and making sure that the biology and the phenotype versus the phenotype of the therapeutic area we're going, the phenotype is matched with the biological dysfunction, making that connection between phenotype and biological dysfunction, understanding that from the beginning, going after that biological function, and then knowing exactly what phenotype, what type of patients you want to enroll in phase 1, phase 2. That's a problem that I think, again, has been I wouldn't say ignored. I think the problem has been the complexity of biology, the lack of data. But I think right now we are at a time where EMR data sets, multi-omic analysis of patients, we are now getting to the point where we can start to connect phenotype with biological dysfunction in the beginning when we're doing target selection. That it's target selection, which is a weird thing right in the beginning. That's the thing that's going to ultimately change our success rates in the clinic. And that's where we need to get better, not just on producing new or better molecules, but making sure that we de-risk the biology in the beginning.
Yeah. No, Brandon, that's a really good point. I was thinking about some companies I covered, smaller companies a few years ago that were working on a reversible BTK inhibitor in a certain kind of cancer. One company got bought by $2 billion, and the other company, unfortunately, didn't work. And the difference was, I remember the chief medical officer of the company that got bought, remember him telling me very distinctly once, he said, Hartaj, we have an X-ray image of our drug and it fits perfectly into the binding clamp, right? The other company never did, and that should have been, a signal to me. That was a learning, But I think I would imagine that computational method should be able to de-risk some of that, right? If companies use that more, I would imagine. Right?
Yeah, absolutely.
Right. Xavi, maybe just kind of going to you, if you can talk a little bit about specifically, what it is that GAIN does that's more, that you think you do better than companies out there, and that kind of gives you an edge as a smaller biotech company.
Yeah. So actually, exactly what Brandon was saying. I mean, our lead project that is going to go into the clinic very soon. What we have is, you have a defect in the protein, and this protein doesn't work, and then this can cause either, depending on if you have both alleles mutated or only one of them, it can cause either Gaucher or it can cause Parkinson. But we have been developing for the last 30 years molecules that just break proteins, right? That they make the protein stop working. And actually, one of the revolutions here as well is that with all the data we have, with all the knowledge, we can go for completely different mechanisms of action. Now, there's a lot of excitement about proximity pharmacology, like targeted protein degradation and so on. But actually, what we have done is actually try to fix the protein, right? The protein that doesn't work, now you bind to it. It's called like a pharmacological chaperone, so actually binds to it, and it makes it stable. It works again, and then voila, you have and it's a wonderful connection between the target and the phenotype, which it's just you have to figure out a way of making it work again, right? So that's the type of thing that you can achieve when you go for this non-standard mechanism of action, and you go for allosteric binding sites. And again, there's no data, so you have to generate your own data, and then you can you have to do that the scale, and be able to screen billions and soon trillions of molecules, and find the right one, and then again interface with the biology, because sometimes some molecules are perfect in many aspects, but then don't give the result there, right? So that's also the hard part of drug discovery, is where it really is discovery and not design. But we can now design things that weren't possible before. People didn't dare to do before.
Yeah.
And I think that's, we have, if you look at our pipeline, we have 7 programs, 6 different proteins, and in all cases, we have actually been able to find molecules that bind allosterically on sites that people didn't think that protein could bind a small molecule there, and that gives us a different effect that we can use for drug discovery. Right, for therapeutic purposes.
Yeah, and Xavi, also the platform approach here would be, we cover Moderna, for example, right? And for example, BioNTech, they talk about how they can go from a sequence of a COVID-19 or some variation of SARS-CoV-2, and then go to a vaccine in about 6 weeks and go into the clinic in 6 weeks, right? Used to take almost a year, right? With their mRNA approach. Of course, they're using mRNA. You can do the same thing, right? You can speed up that process, right? That when you have a protein, you can use, for example, like an AlphaFold protein, do your experiments and come up with far more hits, right, to test in vitro, essentially, right?
Absolutely. And on this early part, we can decide if the molecule offers this sort of new opportunities I was mentioning before. In a couple of weeks, we can identify molecules that could bind to that new site that we have found in about 1 month, and then you do the test, and in a matter of maybe in total 2 months, you can have molecules that have these new functions. So that is really enabling. I mean, that's really, and actually we have extra capacity, right? Because we do that so quickly. We generate our own, we work on our own targets, and then we have to optimize the molecules. So we're actually actively looking for partnerships, and then actually we do have some partnerships on using the technology for other people.
Yep. Yep. And then, Xavi, I just want to be clear. For example, like Vertex, a company we cover, right? They use a small molecule approach to essentially stabilize the CFTR protein and use sweat chloride as a biomarker. And they were able to get 4 different products to the clinic within 3 to 4 years, which is a record almost every time. With enough money and hopefully resources, intellectual and physical, GAIN is trying to kind of do something analogous to that, right? Something like that, I would imagine.
It's the same mechanism of action and in different proteins. Yeah. So you try to fix the protein and make it work again. Yes,
right.
Exactly.
Yeah. No, fantastic. And then we just got some minutes left here. I'll rotate between the 3 gentlemen. Brandon, starting with you in terms of being a public market analyst, and we vet private companies, a tough funding environment. Brandon, what are the 1 or 2 things you would request us in the public markets to think about when we vet companies that are using computational methodologies, whether it's tools or they're discovering drugs, et cetera, to really give them understand their business and hopefully give them a better valuation?
Yeah. Again, I would, right. So one of the things that's often happens in this field is a focus on the lead candidate, right? Again, unfortunately, we joke that investors can only count to 1, right? You might have a pipeline of five programs, but the one that matters is the one that's ahead, and that's basically the valuation that the company gets. And so, I would say. Take some time to understand the potential of the platform and the depth of the pipeline and how those things are synergistic and can expand on the value. So I guess that would be the thing I would ask. And as Jason pointed out earlier, you asked a very critical question: is how do you build a company around a computational platform? And there, I think that's still a huge challenge, because how much do you value the platform? How much do you value the pipeline? And I think we really need to start thinking about a hybrid approach where it's not just the lead candidate in the pipeline, it's the full pipeline, but it's also how is that platform de-risking that pipeline, right? And understanding how that translates to probability of success in getting those compounds to market.
And then Jason, I'll just kind of go to you also. I mean, you're kind of also right there in the thick of it. Public market analysts can be both a whipping boy and or a whipping man, or and also useful at the right time. What advice would you give us, when we're thinking about companies that are kind of coming through the pipe from private companies, where you think there's a misunderstanding, a disconnect between the private and the public approaches for computer-aided sort of companies?
Well, I would really look at the platform and look at how extensible it is. Can the platform grow? Meaning, can it add complexity? Can it scale? If we were to give a company access to an exascale machine, would it be of value, right? That sort of thing. I think the other thing is really important is to understand what is its sweet spot. Is the company's goal to take it to commercialization, or is it to partner it out at phase 1 or phase 2 where you see a really strong value inflection point, right? Yep. There's a massive amount of cost in standing up a commercialization program. You know this. So maybe that's not the goal of every company to be a Merck, Pfizer, Moderna, right? Maybe it needs to say, look, our goal is to get things to phase 1, phase 2, and understand why are they going to be the greatest at that space. Yep. And then I think the the other thing I would really look at is really smart partnerships.
Mhm.
It's just not, I don't think it's good enough just to send your drug out anymore. I think you really have to think about who you're working with. And again, kind of pushing back toward that Google, Apple model of that ecosystem, that tier 1, tier 2, tier 3 supplier model. You know, your partners are actually you want to build a good neighborhood to build your drugs using your approach. And are they doing that?
Yeah. No, that's fantastic advice. I mean, I always tell people the NBI index is 300 companies, right? Versus at 1 trillion versus one Google at 1 trillion or more, and Apple. We got, biotech is going to grow. So I think it's better we try to solve some of these, what could become roadblocks or, down the road now, and think about them now than down the road, right? Xavi, I'll just end with you. Maybe, what would you like investors to think about GAIN, and what do you think is the greatest misunderstanding about GAIN, and why should they be looking at GAIN?
Well, hopefully I've tried to explain. We can do things that I haven't seen anybody else doing, right? So it's not just doing it faster. It's actually doing something that other people can do or haven't been able to do, and the connection between the mechanism of action, what these molecules do, and the disease is very straightforward, right? And it's just that this is the reverse of what people have been doing before, and therefore that should translate into a better prediction of what's going to be the outcome in the clinic, right? There's a more direct relationship between the disease and the phenotype, right? So the phenotype and what your molecule is doing. So yeah, I hope they will see that the molecules actually have a huge potential, and that the platform can actually generate many more molecules like this on a very broad range of disease areas, not just rare diseases or CNS, but also in oncology and other areas.
Yep. No, thank you, Xavi. Thanks for everybody listening. I just mentioned that GAIN has two big things to look forward to this year. One being a product in the clinic, and then number two, hopefully some partnerships. As Jason said, the quality of the partnerships matter. And we will look forward to keeping this conversation going. And hopefully, as Brandon and Jason and I wish for, an R&D day for GAIN in Barcelona.
Yes.
Thanks a lot, everybody.
Take care, everyone.
Bye.
Bye.
All right. Bye.